Welcome everyone to our GoFundMe page. This fundraiser is designed to bring in funding to start a phase 0 clinical trial. Hoag Cancer Center prides itself on using the latest and the best technologies to help our patients, but sometimes we need to push the needle beyond where it currently lies to advance technology. I'd like to describe in this slideshow what a clinical trial is, why this trial is unique, and how anyone can contribute to make a difference in cancer mortality rate. I'm going to discuss our trial, how to financially support our work, who our volunteers are, and what a cancer vaccine actually is. A clinical trial is a test to assess whether a drug or treatment is safe, that's phase 1, and whether it's efficacious, that's phase 2. In this case, we're going to be doing a phase 0. We're monitoring patients who have colorectal cancer and are already being treated, and we're going to look at their immune response. We're going to monitor 200 patients with colon cancer for 6 weeks during their treatment for any indication they might respond to a cancer vaccine. We're also going to monitor 200 healthy volunteers for the same thing. It's possible that there are immune responses that show up in colon cancer patients that don't show up in healthy people or in people that don't have any indication of colon cancer. If that were the case, then we have some potentials for creating a vaccine. How do you contribute? Many clinical trials have potentially a large pharmaceutical company or a CRO that's their sponsor. This trial makes use of a peptide vaccine, which is never going to be a commercial blockbuster like Herceptin or Keytruda. Whether it's an mRNA vaccine or a peptide vaccine, costs to make a vaccine are generally low. They're much lower than making a small molecule chemotherapeutic. Side effects are often very minimal compared to chemotherapeutics, but it is very unlikely that any cancer vaccine will be sold for hundreds of thousands of dollars. A vaccine by nature is inexpensive, and therefore the commercial market for a vaccine is limited. So these facts tend to reduce the enthusiasm of large companies to sponsor vaccine trials that target cancer. But private fundraising can be a primary resource to enable anyone to contribute. Now, who are our volunteers? We are going to bring in at least 200 colorectal cancer patients. If we expand the trial based on the funding that we get, that would be great. If we have to limit the trial based on the funding that we don't get, we can still shrink our trial and generally can make do with as few as 50 patients. We're going to try to get at least 200 non-cancer volunteers who are going to be donating blood, and we can compare the two. Every person is going to receive all the information that we learn. It's possible that our hypothesis is not tenable, our sample size is too small, or more likely, the variability of immune response is so high that no common vaccine would work. All of our volunteers know this first trial is only going to produce data for further trials. Very little of this phase 0 information is useful for a treatment, but the phase 1 trial that we're going to start as soon as we get information and approval will be allowing for active treatments, real vaccines. But it's based on the information we learn from this phase 0. Cancer vaccines basically work because cancers produce mistakes in DNA. Otherwise, they wouldn't really be a cancer. Melanoma and other cancers like non-small cell carcinoma are potentially caused by environmental effects. Some are caused by inherited defects. Some are spontaneous and just accumulate over time until a cell becomes cancerous. It's just bad luck. A frame shift mistake is a good place to start for a vaccine. A frame shift occurs when one nucleotide is either inserted or deleted, and it changes the reading frame. A reading frame is 3 bases, 3 nucleotides. Those 3 nucleotides correspond to an amino acid. If you shift that off by 1, whether you take away a nucleotide or you add a nucleotide, that 3-base triplet is no longer what was designed. So it starts throwing in amino acids that were never designed to be in a protein. So these nonsense peptides sometimes wind up on the surface of a tumor cell. Tumor cell puts them out there, the immune response, the immune system sees them, and it tries to destroy that cell because the human immune system sees that peptide as non-self. So when you combine this with a checkpoint inhibitor, you turn the brakes off of the immune system, and the frameshift vaccine can rev up the immune system and cause it to destroy cancer cells. And the good part about this is even metastatic cells are susceptible to killing no matter where they are in your body. So our current study requires a minimum amount of funding to evaluate peptide antigens. We have to print our microarrays, we have to generate peptides, we have to collect blood, process samples. This is a cost that's inherent in that test. But every sample we process is a new piece of data and it adds to our database of knowledge. Our phase 1 clinical trial absolutely relies on the data we collect in this phase 0 trial to ensure that we're treating patients with vaccines that are most likely to work. Can we predict vaccines from public data? Can we predict antigens that patients may respond to from public data without having to do sequencing ourselves? Yes, we can. Now, Hoag has thousands of patients that we can use for our vaccine prediction, but there's also enormous amounts of data in the public domain. DNA-seq and RNA-seq data is available from CBioportal and from NCI, from many public repositories. Thousands and thousands of cases exist in the public domain. Now, clinical evidence, on those graphs that you see below, suggests that there are some frame shifts that convey protection against cancer. The orange line is one patient or several patients, in some examples, where that frame shift mutation exists. And the difference between survival of that orange line and the blue line, where people don't have that mutation, is significant. Now, whether that single mutation is responsible for protecting that person or those people from cancer is not entirely clear, but the evidence is mounting that these frameshift mutations in the right environment, and when you use Pembro or Keytruda or a checkpoint inhibitor to allow the immune system to rev up and to attack tumor cells, that evidence plus these frame shifts plus clinical trials are very suggestive that this is a highly likely and very low side effect method to produce a drug, a treatment for cancer. Now, how do neoantigens work? Well, a neoantigen is a mistake. As I mentioned, a frame shift is one type of mistake, where a non-self peptide is produced. And that non-self peptide is presented on the surface of a cell, whether it's MHC1 or MHC2. There are multiple different ways and very complicated ways of presenting a cell to the immune system. The patients, their own genotype for their HLA molecules can cause them to either see or not see a particular peptide. That is part of the variability in how cancer vaccines either do or don't work. So the immunology behind these trials, as simple as it may seem on the surface, can be extremely complex. So we pick peptides based on our information. We manufacture them, we vial them, and for our phase 1 trial, we'll be giving them as a prime and several boosts to our patients. And in many cases, the CD4, CD8s are revved up. You get common protection, you get T helper cells, you get B cells, all combining to attack and kill the tumor cell. A vaccine trial from several years ago showed just a few of these frameshift vaccines were enough to cause protection in cancer patients who were Lynch syndrome. They had colorectal cancer, they had mutations that led to frameshifts, but they were able to be protected by several different frameshift peptides. In some cases, the frameshift peptides that the clinician used didn't have any pre-existing immune response. They were completely naive. The patient did not have any antibodies, didn't have any T cells to these antigens. But in some cases, there were pre-vaccine antibody responses. This is exactly what we're looking for in our trial. We're going to see whether or not patients who do not have any vaccines have a pre-existing immunological response to these frameshift antigens. The last few years have led to an explosion of papers, manuscripts that have shown peptide-based vaccines for cancer and frameshift neoantigens are really becoming commonplace in clinical trials. A few years ago, no clinical trials had been used for cancer therapeutic vaccines, and now there are over 617 in clinicaltrials.gov. This is a major turning point. This is the point where we are going to start using and harnessing the immune system to really hone in and precisely kill tumor cells for any type of cancer. Arizona State and the Biodesign Institute have been working on peptide vaccines for many, many years, well before it was accepted that these would even have a chance of working. So the mouse studies and the dog studies have gone on to show that very successful prophylactic and therapeutic uses of cancer vaccines. When we look at common neoantigens from frame shifts, we start to see that there are dozens and dozens of cases where the same patient has exactly the same frame shift as another. Now, with a pool of 10 to 15 different antigens, we expect that our phase 1 clinical trial can likely work on 50 to 60% of all colorectal cancer patients, just based on statistics. Checkpoint inhibitors and peptide vaccines are not always guaranteed to work for every single patient. Now, at Arizona State University, we developed a method to use serology to test for whether or not a checkpoint inhibitor was likely to work and whether a vaccine was likely to work for a given tumor. This information is very valuable for screening patients to include in our cancer vaccine phase 1 trial. If we knew ahead of time that a cancer patient was going to respond or had a high likelihood of responding and another wouldn't, we would definitely try to enroll the patient that was most likely to respond. This is part of our trial, this is part of our science, this is part of the history of all of this research. And thank you for listening to our pitch. We hope that you will consider funding our proposal, and we will definitely keep every donor up to date with all of our results. Thank you.